All right. One of the things that Step 1 for microbiology loves on the boards are virulence factors. They want to know not just that you can say, you know what, this guy came in and he was coughing up, bloody sputum, he had a fever, he had pneumonia. They want to know that you know how that organism caused disease, okay? So what we're going to do here is really just kind of go over some of the virulence factors that are very, very tested on Step 1 of the USMLE, and we'll talk about them in general here, and I'll give you a few examples. And then, when we get to the actual organisms, we'll cover them again. So I may say later on, for example, for E. coli, in a urinary tract infection, the pili are going to be the most important virulence factor. That may not be the case in E. coli meningitis or E. coli gastroenteritis. So depending on the site of the infection, the important virulence factor is going to change. Okay? So the very first thing we're going to talk about are pili. And pili are basically for attachment. Now, that is really important for the bacteria. If you think about this, let's go back to our friend E. coli. Okay? E. coli, let's say he gets lost, doesn't have his GPS, he gets lost from the colon, he makes his way to the urethra. Okay? If E. coli can't attach, what's going to happen? All right? If it can't attach, what's coming at it? Urine coming at it really fast. If it can't attach to the urethra, it's just going to get flushed away. So pili for E. coli are really important. They have to grasp on. Otherwise, they'll never be able to initiate the infection at that particular site. Okay? In Neisseria gonorrhea, pili are also very important. They're important for attachment. But with gonorrhea, they're a little bit different in the fact that the pili are always kind of changing. So once you make an immune response to the first set of pili, the pili start to change. They're like, dude, you're not catching me, I'm changing. So now, the first immune response you made, not working. For the same strain of gonorrhea, just different pili, which is why you can get chronic type infections with Neisseria gonorrhea. You can have them for long periods of time. That's why we think septic arthritis in a sexually active young individual, because that infection can stay around for a long time because the immune response, as good as it is, has a hard time finding the organism. Some of the other adherence markers you see on here, and teichoic acids are basically pili that are found in gram-positive organisms, okay? So you definitely want to be careful there. If you're asked a question about an important attachment virulence factor in a gram-positive organism, it's not going to be pili, it's going to be a teichoic acid, all right? In addition, we have adhesins, and a pertussis toxin is a good example of an adhesin that also helps with attachment. I always tell my students when I do the live lectures that the IgA proteases, if you are a mucosal pathogen that's worth your weight, you're going to have an IgA protease. Why do you think that is? Because the number one protection at the mucosal surfaces is IgA. If you can degrade it, you are home free, okay? Now, biofilms is the last thing listed here, and biofilms are really kind of neat. I don't know if you've ever been to a concert. Back when I went to concerts, like we were really diehards, we'd get something called a general admission ticket, all right? General admission ticket means you walked in on that floor with 10,000 other people, and you sat there like this. You couldn't move unless the crowd moved. If the crowd moved this way, you were moving this way. And if the crowd moved this way, you were moving this way. And if the crowd was doing something illegal, you were doing something illegal. But guess what? The cops were on the outside of the general admission. So the cops, even though there was all kinds of illegal things going on, couldn't get to you, okay? Think about that when we're talking about a biofilm. A biofilm are a bunch of bacteria that adhere they're packed tight together, okay? They're packed so tight together, the immune system can't get to them. They're all kind of protecting each other. The way that the biofilm works is they're almost like peaks and valleys, and in those valleys are kind of where the food comes in, so they all kind of eat together, drink together, and they protect each other, just like in that general admission at a rock concert. So you probably haven't experienced that, but I can tell you because I live to tell. All right. Probably, in addition to the pili, capsules are one of the most important virulence factors. You really do need to know the organisms that are associated with capsules. There's 2 listed here, strep pneumo and Haemophilus influenza, and I'm going to give you a mnemonic that is not all-encompassing, so it doesn't take every single encapsulated organism, but what it does is it takes the ones that are most commonly tested on the USMLE. Okay, so here we go. Some killers have pretty nice capsules. So some is Streptococcus pneumoniae, killers is Klebsiella pneumoniae, have is Haemophilus influenzae, pretty is Pseudomonas aeruginosa, nice is Neisseria meningitidis, and the C is actually not a bacteria, it's actually Cryptococcus neoformans, which is a fungal agent that we'll talk about a little bit later. That takes probably the most predominant organisms that are encapsulated, so you can remember those. Whenever you are being tested on most of those, surely for strep, Haemophilus, Klebsiella, Neisseria, when you're asked, you're given a vignette. Let's say you have a college-age student that comes down with bacterial meningitis. They're not going to give away the diagnosis. They're going to tell you had an 18-year-old male or female living in a dormitory. They all of a sudden presented with symptoms of a stiff neck, with a fever, and probably vomiting. And they will ask you what the most important virulence factor of the most likely causative agent is. Okay? In that case, it's a capsule. And in general, when you think about meningitis, think about capsules, because meningitis, when we're talking about the meninges, we're talking about a protected area, right? The blood-brain barrier. We're talking about something that's actually protected. So what has to be there to allow it to cross that blood-brain barrier is a capsule. Capsules function by helping those bacteria avoid the immune response. And how do they do that? Okay? What they do is they coat the bacteria, and they're very slippery. So if you've ever picked up a bar of soap in a shower and it slipped out of your hands, that's exactly what a capsule does for the bacteria. The neutrophil, like Pac-Man, you probably don't even know Pac-Man anymore. Pac-Man was an old school video game. Like Pac-Man is a neutrophil going to chew up the bacteria, but it can't grab it. It keeps slipping away because the capsule is really, really slippery. So we say that it's anti-phagocytic, meaning the phagocytes can't catch it. Okay, it's just really too slippery. In addition to capsules, there are other proteins that function in a similar manner, meaning that they're anti-phagocytic. And we have some of those listed here. The M proteins for Strep pyogenes actually are the most important virulence factor really for Strep pyogenes as far as being anti-phagocytic. For Neisseria gonorrhea, for Staph aureus, we have pili and we have protein A that function in an anti-phagocytic manner. Okay? IgA proteases clearly helping you get away from the immune system. And we said that any mucosal pathogen worth its weight is going to have an IgA protease. So you are going to see that as a very common theme as we move through. Siderophores are kind of like a BB gun, okay? Siderophores are like, oh, I'm going to steal iron. Is it probably the most effective? Not really. But also helping avoid immune destruction. Now, antigenic variation, I mentioned already with Neisseria gonorrhea and the pili changing. And I said that because the pili change, you can wind up with these kind of lengthy infections with gonorrhea that can go to septic arthritis, for example. And what we've done here is really just kind of list some of the other organisms that can undergo antigenic variation. One of them is a trypanosome brucei rhodiense, and gambiense, East and West African sleeping sickness. These can also undergo phase variation. So again, once the immune system figures out how to start attacking the organism, they start to change the proteins in their outer coat. So that's a similar theme. And you can see that as well in some of the Enterobacteriaceae. You'll see that, for example, Salmonella has thousands of different capsular serotypes. There are different flagellar serotypes because the flagella can change. And we'll talk about that in a little bit more detail when we get to the Enterobacteriaceae in Chapter 2. Now, surviving intracellularly, how clever is this? Okay, you know, as soon as you get in to the host, you have this whole weaponry and arsenal that are going to be chasing after you called the immune system. So how clever is it to get in to one of those cells and escape the way that that cell could kill you and then hide out in that cell? Okay? There are quite a few agents that can do that, so I'm going to talk about a few of them now, and then we'll talk about basically clinically what you're going to see with some of these types of infections. All right? Probably the one that's most common that you've probably all heard of is TB. All right? Everyone knows TB is a facultative intracellular pathogen. Tuberculosis, being facultative intracellular, gets into the macrophage and then inhibits the fusion of the phagosome and the lysosome, and then basically escapes. So what happens then? Well, the macrophage is mad, all right? It knows it has an infection, and it's inside of the macrophage, and it can't kill it. But that's its job. The macrophage's whole job in life are to kill these things. So what do? They can't kill it, so they will just form a wall around it, seal it up in a granuloma. So one of the things you're going to see commonly with these intracellular types of organisms is you'll see a granulomatous response. And that's going to be very common with the organisms that are listed here. All right, so TB is the first one. Listeria is another one that we're going to talk about that also is very common for the granulomatous response. In fact, when Listeria crosses the placenta, the disease that it causes is granulomatous infantisepticum. It just helps remind you about that granulomatous response and the fact that it's intracellular. So what I'm going to do is show you a little animation of how Listeria kind of avoids killing by the macrophage. So if you look at the depiction of the cell on the slide, you can see basically we're in a macrophage. The organism, the pink organism in here is Listeria. It's in a phagosome. And here's your lysosome. Now, we will talk about in the immunology what happens with phagolysosomal fusion, but for a bacteria, it is a nightmare, okay? We're going to basically throw all kinds of nasty, nasty chemicals at it once that phagosome and lysosome fuse. So what does Listeria do? It's like, oh no, you didn't. I am not going into that phagolysosome. So it escapes the phagosome before the phagosome and the lysosome fuse. And then, as you can see, in the cell, it just starts replicating intracellularly. Listeria, once it does this, can also polymerize actin, and it can propel itself into neighboring cells without actually getting outside of the cell. Now, how clever is that for a lowly prokaryotic bacteria? Pretty good, okay? Now, with TB and Listeria, we're talking about bacterial agents that get inside of phagocytic cells. And phagocytic cells are, of course, the cells that are designed to take up those bacteria, process them, either kill them and or present them to T cells for killing. Invasins are actually proteins that allow non-phagocytic cells to take up bacteria. So now, if an organism has an invasion, like Yersinia pseudotuberculosis, it can stimulate non-phagocytic cells to take it up. Now that's probably even more clever because a non-phagocytic cell is not going to have the killing mechanisms. It doesn't have to escape once it gets in, it just has to get in, okay? So that gives you some idea about the intracellular survival capacities of some of the different organisms we're going to be talking about in Chapter 2. Now, type 3 secretion systems are really kind of interesting. These are basically, if you want to think about it like this, a molecular syringe, so that a bacteria can basically just, bam, inject toxins directly into a host cell without necessarily having to get into that host cell, without it having to bind necessarily to an extracellular receptor. We're just going to inject it directly into a host cell. A lot of the enteric organisms have these type 3 secretion systems. So you can see E. coli and Salmonella and Yersinia, which are all the Enterobacteriaceae. Pseudomonas and also Chlamydia have these type 3 secretion systems. So they're basically for delivery of toxins. Next on the list, and we'll focus on this a little bit more when we start talking about the immunology, is a lot of the times it's not maybe the bacteria itself that's doing damage to the host, it's the immune system, okay? This is very common with, for example, rheumatic fever. In rheumatic fever, what happens? You get a strep infection, and the antibodies that you made to the strep, to the group A strep, now cross-react with the heart, and they start causing carditis, and eventually you'll get rheumatic fever. And we're going to get to this more in immunology, but one of the things they're going to test you on the things that are listed on this slide, they're going to want to know what kind of hypersensitivity reaction that is. That's super high yield for these diseases. So for example, the first bullet point that's listed here, cross-reaction of bacterial-induced antibodies with the tissue, that's a type 2 hypersensitivity because you have antibodies binding directly to a cell or tissue. Okay? Now, we move on to the DTH response. The delayed type hypersensitivity response, what type of hypersensitivity is that? That's a type 4 hypersensitivity, and the granulomatous response that we talked about for those facultative intracellular organisms. Now, the third bullet point, the immune complexes, this one should be easy for you. Anytime you see immune complex, you got to think type 3 hypersensitivity, okay? Post-streptococcal glomerulonephritis, that's very high yield. So you definitely want to know that. You want to know the hypersensitivity, and we'll talk about the mechanism of that when we approach immunology. The last bullet point there is that peptidoglycan being part of the cell wall for bacteria, as well as teichoic acid, is actually chemotactic for neutrophils. Now, generally what you're going to see overall is when you recruit immune cells to any different area, and in this particular case, you're recruiting, let's say you have Streptococcus pneumonia. You have streptococcal pneumonia. The peptidoglycan recruits neutrophils. You'll find them in the sputum, okay? But what did I tell you about the capsule? Neutrophils are going to be going. They're going to try to attack the encapsulated organism. They're not going to be able to do it. They're so frustrated. God knows how far they traveled. They just got into to the lungs, and now they're trying to help you clear that bacteria, and they can't do it. So they get overworked, they get angry, and they start releasing some of those bad chemicals and can actually contribute to some of the tissue damage that you can see that's immune-mediated. Okay, so the immune system also very commonly causes damage. And you'll see, for example, in the case of tuberculosis, that organism really has no virulence factors. The entire disease is based on the immune response to that organism. So very important mechanism of virulence. Okay, physical damage, there's not much here. It's pretty straightforward. If you're too big, you're going to start to block things, especially in a fixed space. So for example, in meningitis, we worry about meningitis because we have all this swelling in a fixed space. And of course, if that goes on too long, we're going to start to get death of the tissue, which clearly is not a good thing. So that's a very common example. If you have an overwhelming ascarid infection, I would be a Ascarid if I had an Ascarid infection. But if you had an overwhelming infection with Ascaris lumbricoides, which is the largest roundworm, it can actually block the bile duct. It can actually block the gut if the worm burden is really high. So the physical size can cause damage as well. Amoeba histolytica also can cause physical damage because it directly invades the tissue, okay, and can cause damage then. So physical damage is pretty straightforward, not much there. Most of that is just kind of knowing about the organism. For example, Ascaris, it's the largest organism we're going to be talking about. So clearly, if you get too many of them in a fixed site, it can cause damage.